This little microphone makes it feel like a TED Talk for me, so I hope it's at least that exciting. I'm a Texan native, so this is a really wonderful opportunity to have evolution right here in my home state. So today I'm going to be talking about two chapters of my dissertation research. I'm a PhD student at the University of Texas at Arlington. I'm in Mac Fujita's lab, and I primarily work with these absolutely fantastic animals, the tea-ed lizard, Aspidoscelis tesselata. And what's so cool about it is that it is a hybrid species, which is already cool, but it's also parthenogenetic, and there are no males in the species, and to our knowledge, no males are necessary in order for this animal to reproduce. So an F1 hybrid produces eggs, they're fertile, and ta-da, you have basically a whole new species in an F1. Something that I find really interesting in this species, other than it being hybrid, is that it's quite variable. So I'm just pointing out on this slide the two species that are thought to be the parental species, you could say. On the left here we have Aspidoscelis gularis, and it's thought to have been the male in this crossing, or the father, if you want to think about it that way. And on the right you have Aspidoscelis marmorata, thought to be the maternal or mother species. And right smack dab in the middle, you have Aspidoscelis tesselata, the all-female species. If you look very carefully at their scales, you'll notice some characters from both species. I don't have the time to get into that. So that variation I talked about has been long known. You can see from this citation from the 60s by Richard Zweifel at Harvard, I believe at the time. He describes what he called 6 pattern classes. This was basically his reluctance to call these different things different species, and he gave them letters, forever complicating the taxonomy of this genus. If you're at all familiar with it, you'll know that to be the case. So we now know that there are actually 3 species collectively at that time called Aspidoscelis tesselata. The 2 on the left, pattern classes A and B, we now know are triploid, and our second hybrids with another species, I have no time to talk about that, even though it's really cool. And then the 4 on the right here, B through F, are, at one point they were called Aspidoscelis tesselata, and then later in 1973, James Scudday renamed one of these groups Aspidoscelis dixoni. I will talk more about that guy in a minute, but what is worth noting, these guys are hybrid, these 3, girls really, and they're diploid, and they have 1 copy of chromosomes from each parent species. So my research questions here are twofold. How many origins, because they're hybrids, are there for this Aspidoscelis tesselata group? And then also, what is that dixoni thing? What makes it so different that it's called its own species, if it even is that? And these are the sources of evidence I'm going to present today. So to begin with, these animals are found in the American Southwest, so this should look familiar. You're about there. My hand could stay steady enough. So the 3 species, again, Gularis, which is the father species, Marmorata, the mother species, and Tesselata, the hybrid. And these colors I'm going to use throughout the rest of my presentation. I'll try and help you keep track. So over here, these are samples that I'm using for genetic work later. You can see, if you kind of combine in your mind, that these ranges somewhat overlap, with Tesselata having a more northwest distribution compared to the other 2, Marmorata extending a little bit into Mexico across the Rio Grande River, and then Gularis extending quite a ways down into central Mexico. So the first technique that we use to identify the number of origins of Aspidoscelis tesselata, this was done in collaboration with another graduate student in our lab, Danielle, who's in the audience back there. What you're looking at, again, the same colors. These are ecological niches projected onto geographic space, and each species has its own color, and there's some different colors for where the species niches overlap in geographic space. So again, in red we have the father species, Gularis. This is 6,000 years ago. Tesselata, the hybrid, is still farther north, mostly, and Marmorata is over here to the east. And what's really cool about this figure, in my eyes, is that in this dashed patterning here, this is where all 3 species niches overlap in geographic space. And if you look carefully, there's 2 main regions where this is happening, which is very exciting, and I guess I should mention the null expectation, at least from the research, is that there's a single origin of this parthenogenetic species. So this is very interesting and something we looked further into. If you project farther back in time to the last glacial maximum, it looks like there's really only one area where there's significant overlap between the three species. And then interestingly, if you go back pretty far to an area that was much colder, two of these species barely have any projected habitat suitability in geographic space. I don't honestly believe there's much to this level of overlap way over here, especially in Baja, which is geographically separated by quite a bit. So it implies that this hybrid species is probably pretty young, so less than at least 120,000 years old. So as for the genetic data, I collected double digest restriction-associated DNA sequencing summarized on the right here. I'm going to focus mainly in terms of genetics on this other data set where we used entire mitochondrial genomes, and the way we're doing this, you can speak with me afterwards if you'd like some details, but we're digesting away linear DNA, and that leaves you only with circularized DNA. There's going to be some bacteria in there, but most of it's mitochondria. And then we linearize that, amplify it, and sequence it using standard Illumina library protocols. And since there's no TID reference mitochondrial genome, we created a pseudo-reference genome. That had to be our first step. After trimming and demultiplexing, etc., the reads, we used the programs Abyss and Cap3 and fed that into BWA-NEM to create a pseudo-reference genome. This is Aspidoscelis sexlineata species. And that was used in some cases to bolster the efficiency of assembly for the remaining individuals. All others were just done in Abyss and Cap3. We then did a sanity check by checking for contamination using BLAST, and we annotated the genes using an online web server called Mitos, which is super cool. We then aligned the sequences in muscle, fed that into Genius to get the gene regions partitioned by gene, and then fed that into Mr. Bayes. So I also used Beast, but I'm only going to show you the Mr. Bayes results today for sake of time. This is a mitochondrial genome tree of approximately 40 individuals. What you're seeing, I'm going to guide you from bottom to top here. Again, the colors are consistent with the previous figures. This is an outgroup from another family of lizards. It's a Lacertid podarcis. Over here, we have a group of species that are all in that are all associated with the paternal species, Aspidoscelis pilaris. The branch links are fairly long here because these are three species, arguably four. And then over here in blue, in this light green color, this to me is where the interesting biology is happening. You're seeing Aspidoscelis marmorata, the maternal species, has very shallow population structure, at least based on mitochondrial data. And then from within that, you can see a single origin for the mitochondria of Aspidoscelis tesselata, the parthenogenetic species. Now that's so clustered together, it's hard to see. Now this is a cladogram then, and you can see with this arrow, these are all in green here, the hybrid species, and it's only popping out at one place on this tree. Something that's interesting to me personally, these two individuals right here at that split point are the only two I have a particular subspecies of marmorata. I haven't delved into subspecies, but those are found very close to the Rio Grande River, and actually very close to one of those two regions we recovered in Danielle's ecological niche modeling work. So, to recap, I've talked a lot about these three, it's no longer pointing, there we go. I talked a lot about these three pattern classes, which we now call Aspidoscelis tesselata, but I haven't really commented on this sucker over here, dixoni. So I'd like to spend a moment on that. So dixoni is also a diploid hybrid, and the same species are thought to be the parent species for this also hybrid, also parthenogenetic lineage. Now what's weird about it is that if you squint and look at these things hard enough and long enough, and other people have, you can convince yourself that they are something that's actually quite different, because they just look different than the tessellata. They also have a couple more scales on the back. This is important to people who study these things. Okay, but this species also has phenotypic variation in it. Again, this is more of a, once you're familiar with it, you see it kind of thing. But they're giving, again, pattern classes A, B, and C. So I think the coolest thing about them is that they have a really, really weird range. They're found in incredibly restricted habitats, just this dixoni group. Over here, pattern class C is found in the southwestern portion of New Mexico, in the Presention Mountains, and then A and B are found in the Chinati Mountains in far west Texas. So where does it go in this tree? Well, based on the mitochondria, it fits firmly within Aspidoscelis tesselata. This implies that Aspidoscelis tesselata, if you believe that it's a species, and not everyone believes that, it's paraphyletic with that including dixoni. So this suggests that dixoni is not a valid, separate evolutionary unit. If I have the time, I think I do, I'd like to describe very briefly work that is done in association with two co-authors on this talk, James Cordes and James Walker, in which we've discovered another population way up here. And if you know, Texas is the second largest state in the United States. This is a huge distance, almost 400 miles, and maybe, what is that, 600 kilometers-ish? And they've never been found in between. So I'm working to help them describe this individual right here. This is from Palidero Canyon State Park. If you have super eagle eye vision, you notice that one patch of its skin appears to be rotated. That's not an accident. This was work done by James Cordes, the collaborator. What he's done here is he's actually transferred a piece of skin from Aspidoscelis tesselata onto dixoni, this individual, well, we think it's dixoni. And the idea is, just like when you're doing an organ transplant, you have to be very closely related to that organism in order for it to, quote-unquote, take and not turn into a scab. This is a skin histocompatibility test. Since it took, we have suggestions that this is a very closely related lineage. Okay, so in summary, there appear to be multiple origins of Aspidoscelis tesselata. However, only one mitochondrial origin, and also Aspidoscelis dixoni is subsumed by Aspidoscelis tesselata. As far as explaining this pattern, what I think is happening is that either I haven't sampled enough Aspidoscelis marmorata from the Mexico portion of the range, or, and this is supported by the shallow mitochondrial topology, there was probably a population restriction at some point, and now we're only able to sample one of the two mitochondrial haplotypes, which are the maternal origins. With that, I'd like to thank many, many people. I don't have time to list all the names, but I'd like to point out Jessica here. She helped me do a lot of field work, and then my funding sources, SSE and NSF. And with that, I can take any questions.